So we can get started. So thanks everyone for coming to our August webinar. We're going to start having these monthly again now that school is back in session usually, and people are off vacation. So we're going to start resuming these for the rest of the year. So thank you for attending. So today we're going to be talking about determining polygenic risk score. So here's a brief overview of the agenda. So we're going to do a really quick meet your moderators and an overview of the overview of AMPD and the AMPD community. And then we're going to get into Mary's notebooks. And if you have any questions, please feel free to either save your questions for the end or put them in the chat. We will pause in between the two different notebooks in case there are questions pertaining to a specific notebook. But that way we don't interrupt the flow of a notebook as Mary is going through it. So we do have a survey that I can send out in the chat in a little bit. So as you're going through this webinar, if you could please fill out the survey, that would help us out a lot, and get these more tailored towards what you're looking for. Yesterday we had sent out a Terra workspace link for any of those who have tier 2 access. So this is the first time we've done this for a webinar, so for the public webinar. So if you have tier 2 access, you should now have access to the workspace, the webinar workspace. And then we did also send out a PDF of the code. So thank you, Mary, for that suggestion. So those of you who don't have tier 2 access, you now have a PDF of the notebooks that Mary's going to be going through for your reference, if you want to follow along with those as well. And so let's get into it. So we have Mary, our main presenter. So thank you, Mary, for joining us. My name is Victoria Dardov. I'm a data scientist at Technome, and we also have with us Willie and Dave. So Willie and Dave, if you guys want to give a quick sentence about yourself.
Yeah, hi everyone. I'm Willie Nojer-Parnoto. I'm a solutions engineer at Verily, and I've been working with Technome on the AMPD project for several years now. So happy to answer any questions about Terra, AMPD, anything you have in mind.
Thank you.
And I'm Dave Vismer. I'm a senior engineer at Technome, and I'm part of the admin support team for AMPD. So if you have any questions, trouble accessing anything, or need any help with notebooks or anything like that, I'm happy to help.
Awesome. Thank you. So real brief overview of what is Parkinson's disease. So if you're already at this webinar, you probably know a lot about this disease, but I was just going to do a really brief overview. So it's a devastating neurodegenerative disease caused mainly by malfunction of dopaminergic neurons. So there's a decrease in dopamine signaling. And so that leads to a majority or a lot of both motor and non-motor impairments. And as disease progresses, there are no cures for this. So the only thing that we're able to do is manage severity of symptoms with treatment. So we have progression of disease, and as disease progresses, we're able to tamper the severity of the symptoms with treatment, but currently there's no cure. So AMPD was kind of came out of this and is a private public partnership with both government, nonprofit, and industry partners. And AMP aims to validate and identify the most promising biological targets for therapeutics so that hopefully we can not only just find treatments for PD patients, but also cures. And the AMPD community has a wealth of resources, so including this webinar, but we also have a lot of visualization tools. So we have the AMPD Data Explorer and a few other tools as well, Gene Explorer. We also have a lot of notebooks and workspaces that are generated as resources for the community. So those are available to those with tier 2 access. And then we also have the AMPD community. So we have individuals from the AMPD community who are contributing resources and notebooks to AMPD so that it could benefit individuals like you. And yeah, we also have this webinar to showcase a lot of these community-built workspaces and notebooks. And so with that, so today is August 10th, and we have our August 10th webinar. And then our next one is September 14th, and it's going to be getting started with proteomics. So if you are interested in that and you're not on the invite list, please feel free to let me know, and I can add you. And with that, I can hand it over to Mary, and Mary can go through her slides and start discussing and doing the walkthrough of her workspaces.
Thanks, Victoria. Let me just go ahead and share my screen.
Yep, perfect. Thank you.
So we can, we'll pick back up where we left off. I'm Mary Macarius. If you haven't met me, I am a pre-doctoral student. Just means I'm in the middle of getting my PhD right now. I work under Dr. Andy Singleton at the National Institutes on Aging. So I'm actually based here in DC most of the time, and I'm getting my degree through the University College London. So if you haven't met me, hello. And I come from mostly a genetics background, and that's why I'll be highlighting a couple of things today. For this webinar, we're going to be going over two different notebooks, and I thought it was really crucial, before we get to PRS, a really in-depth look at exploring the clinical information that AMP-PD has to offer. AMP-PD is really unlike most initiatives out there. They spend a really long time trying to harmonize all the data across different cohorts so that you as the user, when you come in, it's a lot easier to pick individuals based on a cohort of interest, maybe a couple of genetic determinants that you're interested in for your analysis question. So we'll go through a couple of things here, mostly pulling phenotype of interest, exploring a little bit about the race, education levels, age that's available on AMP, and making something that we call a covariate file. For everyone who's already dipped their toes into genetics research, a covariate file are just these, it's one big text file or CSV that includes features you want to correct your models for. This could be for your machine learning models, regression models, what have you, but these are known confounders that you want to correct for. So I'll be going through that notebook first. And that's a pretty lightweight notebook, but given the wealth of information that AMP makes available, it's not going to be exhaustive by any means. It is just going to give you a very, it's in-depth, but not an exhaustive look into what AMP makes available. So we'll go through the first notebook. The second notebook is mostly, if you're already interested in AMP and you already have explored all of these files, maybe this one's of more interest to you, where we're calculating the polygenic risk score, or PRS for short. This is the figure, one of the figures we'll be making in that notebook, and it's stratified by the disease status, so healthy control, positive PD case, or if you're a prodromal, a SWED, or genetically enriched for known mutations in GBA or LRRK2, or if you have these mutations in those genes, but you're unaffected, but you're not necessarily a healthy control. So what is a PRS? I wanted to talk about that real quick. I think there's a lot of misconceptions out there about what a PRS can and can't do. PRS is a subtype of a genetic risk score, and it's used to evaluate one's overall risk for the predisposition of disease. We're focusing on Parkinson's disease, of course. So it's a set of genetic loci that we have found through genome-wide association studies, a GWAS for short, that are involved in cumulatively contributing to the risk of PD. So this includes the effects of both protective and risk variants. Predominantly risk variants, but it does take into account protective variants as well. So a PRS calculation really compares individual level genotypes and phenotype data to a larger collection of weighted alleles, which we get from our GWAS. And it really indicates an individual's, to an individual, their susceptibility based on the alleles or their genetic predisposition that they might have. So you'll either have a low PRS score or a high PRS score, and most people, as evidenced by this distribution, kind of fall somewhere in the middle. The higher your PRS, the more likely you are to have the disease. The limitations of a PRS. PRS are really only able to show correlations and not causation. The genetic risk is not going to be the only determinant if you are going to have Parkinson's disease or if you're at risk for PD, because PD is such a complex disorder that has multiple it's a really multifaceted, complex disorder. So depending on the strategy that you use to calculate PRS, a couple of things can contribute to this bias. So if you do some sort of linkage disequilibrium-based pruning, that might bias your PRS. So it's always good to keep that in the back of your mind when you're reading papers that calculate PRS. Building a PRS will also have discrepancies due to ethnic backgrounds. I will mention this a couple of times as we go through the notebook. For this particular example that we're going through today, we're really leveraging the largest meta genome-wide association study done on individuals with European ancestry, so predominantly white individuals. AMP-PD also predominantly has white individuals in their cohorts, which is great, but it just means that for individuals that are not of that ancestral background, you need to take the PRS with a grain of salt, and it's most likely inaccurate for those individuals. Since disease-associated alleles can have different frequencies depending on which population you come from. A couple of considerations when we run this notebook. This notebook is really meant to serve as a guide for you, an example on how to run a PRS. We don't remove related individuals, and that's an important caveat to mention. If you are going to use PRS for some sort of regression or predictive modeling, you would want to remove any related individuals from your data first. We use the largest European PD GWAS, which I just mentioned. So it's important to note those would only be relevant if you are calculating for individuals of the same ancestral group. We recommend inferring the Ashkenazi Jewish status. We've highlighted this in the manuscript that came out with the first release of AMP-PD, how to do this. That's just because individuals of Ashkenazi Jewish descent have a larger number of rare variants that are enriched in that particular population and can skew your results when it comes to calculating PRS. So calculating your ancestral principal components, PCs for short, I'm sure everyone here is familiar with that. We haven't calculated them for this particular notebook or example, but those are added covariates you would want to adjust your model for. And we go through running a regression with the PRS to predict disease status. So we show an example of this in the notebook, but your final regression, when you run it yourself, would want to adjust for a couple other covariates as well. We'll go through age, sex, and family history, but we won't go through, for example, Ashkenazi Jewish status or the first few principal components. With that, I will transfer over to our first notebook. So like mentioned earlier, if you have tier 2 access, you should be able to access the workspace now. And this is the notebook that we're starting with here. I won't be running it in an interactive virtual machine. I'm just going to scroll through and kind of walk you through what's happening here. If you don't have tier 2 access, I know that the PDFs were sent out a little earlier today, and you're welcome to follow along there. So our first notebook here, maybe I'll pause here in case anyone has any questions before I deep dive into the first notebook. I can't see the chat. Okay. So moving on. So our first notebook here, this is really to go through the clinical information that AMP-PD makes available. So a couple of considerations here. There is going to be missing data with any data set that you come across. So at the very end here, we do calculate the amount of missingness in the features that we've extracted from these notebooks. There are no PCs in this covariate file, like I mentioned earlier, and that's a pretty important component of a covariate file to include. So getting started here, we're going to load. If you've been familiar with the getting started workspaces that AMP-PD makes available, these first couple of cells are just that, where we load in the packages we'll need for Python, and we also develop a couple of functions as well. So these are the workspace variables that we're working in. Depending on what workspace you're in, We are running everything on the latest release of AMP-PD, so that is the 2.5 release. And we print them out right here, our workspace name, billing project, etc. These are the functions that we've developed through the Getting Started Workspaces at AMP-PD. I highly recommend them to copy paste them in whichever notebook you're using. Makes life a little easier to submit little commands that you might need to run. Nothing we're changing here from the Getting Started Workspaces. I've just pasted them here for your convenience. So we're going to go ahead and list out the available files in the buckets. This is a skeleton of the directories AMP-PD makes available if you have Tier 2 release, if you have Tier 2 access. So this first one here is the case control, where they report the latest diagnosis per individual. I think initial diagnosis and latest diagnosis as well, and a couple other things as well. The reason I had put together this notebook in the first place is that there's a lot of clinical information in here, 58 files to be exact, and I just wanted to go through a little bit of them. What AMP-PD also makes available, and if you've been coming to these webinars for a while, you'll have noticed that there's a wealth of transcriptomics and proteomics data as well. Because we are calculating PRS, I wanted to showcase the amount of genetics information AMP-PD makes available. AMP-PD makes whole genome sequences available, which is an incredible resource, and you won't be able to find that most places. Specifically for our next notebook, we'll be focusing on these Plink files. We'll talk about that in the next notebook. So as we're putting together our phenotype file here, we're going to go ahead and try to get the relevant clinical information. That first file, this AMP-PD case control file, will have an overview of the case control data frame. If you are running this interactively, please feel free to uncomment the data frames so you can see the actual files that we're pulling in. Given that we're recording this, I'm not showing you the actual data frames, but rather a high-level overview of what those data frames will give you. So it will output for you the case controller other. The reason that you might want to look at AMP-PD, because it collects a number of cohorts, there are a lot more diseases captured in there than just Parkinson's disease. That's just for something for you to keep in mind, that there are going to be other things in here. For example, individuals who are diagnosed with Parkinsonism, but not necessarily Parkinson's disease. I think there's one person with Alzheimer's in here. And this is just to keep in mind that for your own analysis, to double check which disorder you want to focus on. There is also an LBD cohort with a substantial amount of LBD individuals in there, if that is relevant to your research question. Another question we receive through AMP-PD is, where do these individuals come from? And while there are multiple resources on the AMP-PD website to walk you through which cohorts are available, you can also get them from the prefix of each of the individuals. So this is a quick function to output for you in a separate column the number of individuals, or excuse me, the cohort in which each individual comes from. And you can go ahead and, again, uncomment here. Phenotype, as reported by AMP-PD, is either case control or other. Because we are going to be running statistical tests, we can't really keep them in these categorical ways. We can't keep them as words, so we go ahead and convert those to 1 for control, 2 for positive Parkinson's disease case, and a -9 for everyone else, just to make it easier when we're running our statistical tests. So when you run this, you should find that we have about 4,000 or so healthy controls, 3,000 and 3,500 positive PD cases or idiopathic PD cases as well, and everyone else as an other. This is great. This is all amazing information that we can get. So at this point, we've highlighted how to get the cohort where each individual comes from and coding their information so we can use it for a statistical test. You could stop here, but We're going to go through extracting the enrollment information as well. Like I highlighted in the slides earlier, specifically the PPMI cohort has recruited individuals that have mutations in particular genes that we know to be associated with Parkinson's. We typically would treat those individuals differently from individuals who have idiopathic PD or Parkinson's disease that we can't actually explain exactly why they have PD. And they've made, AMP-PD has made this available in their enrollment CSV in the same clinical bucket as well. So we go ahead and just count in which enrollment each individual comes from. Again, for anything that has been commented here, please feel free to uncomment and look at that data frame. We're going to collect a couple of demographics information as well. Like in the slides I mentioned, we know the Parkinson's disease community as a whole knows that sex, age, education are all obviously involved and play some role in Parkinson's disease. If you have a higher education level, you are less likely to have Parkinson's, so on and so forth. Because this is really crucial to the community, I go ahead and add to the notebook how to pull that information since they are found in different files. This is found in the demographics file, so we go ahead and extract the age at baseline. And for this particular example, we're only extracting the age at baseline that they have showed us at their first visit month, in case it changes. Education level, AMP-PD has been, they have categorical categories for the amount of education an individual has gone through. So we go ahead and code this just, again, a little differently for our statistical tests. So we give 0 if you have no college education at all. We give you a 1 if you have a little bit of college education, and a 2 if you've received any postgraduate education. That's, again, just so we can include it as a feature in our models. For sex, this was also categorical, male, female. So we code those to follow the Planck convention, 1 for male, and 2 for female. Again, so we can include this in our model later. I did highlight this a little earlier, the distribution of individuals that AMP-PD has. So we go ahead and pull race, and we have about 10,000 individuals who are white or of European ancestry, but we also have individuals from other ancestries as well. This is just to keep in mind for your analysis. You most likely wouldn't combine all of these ancestries for one particular analysis. Here we showcase just the breakdown of cases and controls you have by each race that is available at AMP-PD as of the release 2.5. And we clean up this data frame. Again, you can uncomment here, but the data frame would show you the age at baseline that we have kept, the phenotype that we coded for, and the sex information as well. We merged this information with the enrollment arm. Again, the enrollment CSV had information like healthy control, if you were of a genetic recruitment arm, and this is just important to know as well. We go ahead and visualize the age at baseline for cases in particular, and you can see that we've got quite the distribution here, including some early onset individuals. We pull medical information as well. This, for some of the individuals that AMP-PD has captured, we have information such as PD medications these individuals might have taken, smoking history, drinking history as well. So this is all captured. And I just wanted to highlight the amount of information that AMP-PD does end up capturing here. So these are some of the questions we can answer with that information, pending that it's not missing, of course. Is the individual on any PD medication? Is the individual on levodopa specifically? Any dopamine agonists? And was the diagnosis clinically or pathologically confirmed? For most of these, they are going to be clinically confirmed, as opposed to pathologically. And these are just examples on how to do that. Obviously, I think this notebook and the PDF that you have serve more of a reference to you in order to pull and code this information in ways that you can use for your own statistical tests. So I'm going to scroll through this really quick. These are the sort of questions you can ask, what kinds of medication they might be on. We clean this data here. Smoking and drinking history is something else that AMP-PD tries to capture. Depending on the cohort, they might not capture for everybody. Smoking has a protective effect when it comes to Parkinson's disease, and drinking has a negative effect. So if that is important for your particular analysis question, for some of these individuals, that information is captured. And these are just examples of, has the person been drinking recently, and how many drinks have they had, same for smoking as well. And these are just ways to code it, very similarly to the way that we coded education earlier. Zeros, ones, and twos, so we can include them in our model. And a few examples of visualizations in here as well. So I'm just scrolling through this pretty fast, given that it'll depend on really what you need for your own question. So we're cleaning here the data set. Again, you can uncomment here, but the data that we've captured here is the, if you've had alcohol recently, how frequently, have you had tobacco recently, and how frequently. Another important covariate to keep in mind, if you have a close family member with Parkinson's disease, you are at higher risk of having Parkinson's yourself. So this is also captured through AMP-PD. They have a separate family history of Parkinson's disease file that you can load in. They actually, depending on the cohort, it can be quite granular, whether it is your mother, you get it on your paternal or your maternal side. For the purposes of the PRS model, I really just wanted to capture if there is known family history of PD. So we code those as 0 and 1. If you have a relative with PD, you were assigned a 1. If you don't, you were assigned a 0. This is towards the end of the first notebook here. We're just merging and cleaning the data, everything that we've extracted here. The overview of the data broken down by cohort, phenotype, and the latest diagnosis available per cohort. And we also go ahead and calculate missingness. Like I said, as AMP-PD really tries to harmonize across cohorts, not every cohort is capturing each and every part of the information. So it's good to know what you might need to drop for your statistical tests. If you've got a feature with a lot of missingness, it might not be appropriate to run, and it might be dropped altogether. So at the very end here, we've created this table where these are all the features that I've just walked through in this notebook that we capture, how they were coded for, what the ones and twos mean, and the amount of missingness in that particular feature. So for example, education might not have been was not picked up on for a number of these individuals, alcohol, smoking, and this is just important to keep in mind for your own analysis. So you end up creating this file at the very end of this, including the amount of missingness as well. And we go ahead and save these outputs into your bucket, so you can download them to your local computer should you need them, or if you need to transfer it to another workspace for your analysis. And this is just an example of how to do that. At the very end here, I've added a quick example of how to read the file back in whichever notebook you might be using for your analysis. And for this particular file, we'll actually be using in our second notebook as well. That's the end of notebook 1. It really is just an overview of the types of clinical data that might be out there in AMP-PD, how to code for it, how to save your results, and how to check for the amount of missingness, and visualizing a couple of concepts. Do we have any questions here before I move on to our PRS notebook? Okay.
And just as a reminder, if you come up with questions, feel free to put them in the chat. Yeah, and if you have any questions about any of the notebooks, yeah, feel free to put them in the chat or ask at the end of the notebook presentation. But yeah, stellar work, Mary, like really, really stellar work.
Thank you. So I'm going to go ahead and share this second notebook. You also have this as a PDF if you don't have access to this workspace. The considerations and everything that I went through in the slides, they are also at the top here in case you don't remember exactly what I went through with the slides, or if you use this as a reference later on. There are important considerations when it comes to polygenic risk scores, PRSs. They can be calculated in a number of different ways, in a number of more complex ways as well. For this, we really are just using Plink, which is a standard package or program that geneticists use, and it just runs a simple regression. But there are a number of ways you can run a PRS. You can include fixed effects, random effects, additional types of omics into your PRS, and a lot of different papers do a lot of different things. This is just your basic bare bones, how to run a PRS. Again, I did want to mention that there are certain limitations with this particular notebook. We don't account for principal components here. We don't remove any related individuals, for example. We are running on the full data set, but you would want to remove related individuals or anyone of not European ancestry, given that that's the beta effects that we're using today. So we're not adjusting this model in the most comprehensive manner, but still quite, still in a strong manner. I've also added the background of what a PRS is, how is it useful, and the limitations when it comes to other populations. There's a lot of, this is an active area of research actually, and if this is of interest to you, there are a number of tools out there now trying to leverage different kinds of summary statistics in order to run PRSs in other populations. Just as a disclaimer. So the first thing that we're going to do here, again, at the top of your notebook, you have a little outline in case you wanted to jump ahead and look at something in particular. I'm going to go through this sequentially. We're using a particular package here called Table 1. This is just to make one of the tables we have later on. That is not me, that is outside. The first cell that we have here is the packages again. This is the same as the last notebook. Here we have an example of how to load in your, just to see how much space you actually have in your virtual machine. Because we are copying over the Plink files that AmpPD has made available in order to run this analysis, we need substantially more than what the default environment will give you. And so we've tried to make that clear at the top of the notebook here. What specifications you, minimum specifications you'll need. Oh, great. The minimum specifications you'll need in order to run this analysis. It just logged me out of Terra, so I'm just logging back in. Okay. Yep, this is the same piece of code from the last notebook as well. It really just prints out the billing and workspace. These are the same functions that we used, also the same functions in the getting started notebooks. This is, again, the structure of the genetics data that we're using. And we are in particular going to be using the binary files for the Plink 1.9 version that AmpPD has made available. They make it available per chromosome, but they also have an all chromosomes merged version that we'll be using here. This is just listing out all of the files in that particular bucket. Again, they make it available per chromosome, but we are going to be using these top 4 files here, all chromosomes merged. In order to run this analysis, we will be using something called Plink. I've added the website here in case you're unfamiliar with Plink, but it's a common bioinformatics tool. It was developed at Harvard kind of way back when. They still push a lot of updates to it every couple of months. And it's really meant to aid in the analysis of genotype and phenotype analysis. There are two versions to Plink. There's 1.9, which is a bit, I think, more standard. 2.0 only came out fairly recently, within the last 2 or 3 years. We do use both versions here, so we've walked through how to install both here, and it really should only take you a couple of seconds to load it into your workspace, into your virtual machine. Like I said earlier, we're going to be copying over the 1.9 Plink files, the merged 1.9. This walks through how to copy them over to your virtual machine. For this particular example, obviously I'm not in a VM at the moment, but you can uncomment this command. And it takes a little while depending on a number of things really, but it can take up to 20 to 30 minutes to port over all of those files. So if you are running this interactively, this might be the rate limiting step, but I'm going to keep going. So we're going to read in the PD GWAS summary statistics. You can use any GWAS summary statistics you want. Given that the Knowles et al. 2019 paper is the largest meta-analysis in Parkinson's disease, that's what I'm using here today, and it was performed on individuals of European ancestry. So that is important to keep in mind, that these beta effects that we're reading in are of European ancestry. These are the alternate alleles and the beta effects associated with them. For the largest GWAS, meta-GWAS, there are 90 loci associated with Parkinson's disease. So these are the RSIDs, the alternate allele, and the beta effect associated with that RSID. So the first thing we want to do in order to run a PRS is actually extract these variants from our Plink files. We want to extract all the genotypes per individual for these 90 loci that we know are associated with Parkinson's disease. So this is the command to do that. It reads in that merged file that we have copied over to our VM in the earlier step. We extract the variants of interest, those 90 that we just read in here, and we make a smaller Plink file in order to run this PRS. So this is step 1. And we need to inspect the log file or the output file that comes from that particular code chunk. So when we read it out here, there are only 88 variants out of the 90 were found. So there are 2 variants missing in the AMP-PD files that were made available to us. We could stop here. You could calculate the PRS based on these 88, but it wouldn't be quite as accurate as it could be. So we go ahead and investigate the missing variants as to why we're missing 2 of them. AMP-PD also does make available in a separate bucket these VCF files, and you can pull out each variant and see if it passes the quality control pipeline, the GATK pipeline, or if it doesn't pass. This is just a code chunk to show you that we went through, inspected these 2 variants, and saw that they were removed due to excess heterozygosity. Same thing with the second variant that we're missing here. So instead of chucking those away and running our PRS on just 88 variants, we're going to go ahead and replace them using SNPs that are in linkage disequilibrium with the 2 SNPs that are missing. If you're unfamiliar with linkage disequilibrium, or LD for short, it's the concept that one particular SNP gets inherited with another SNP as a part of a haplogroup. So if one is missing, we are going to go ahead and look for a SNP that should have been inherited as a part of that haplogroup. These are missing SNPs right here, the RSIDs. There's a tool called LD proxy where you can go in, submit your SNP, and it will output for you. And I've put the outputs here in this notebook. Output for you other RSIDs that are in LD with the SNP that you put in. So this is for our first SNP that was missing, and this is for our second SNP. These are the outputs that you would get from that website I've linked here. Our files are in HG38, which is important to keep in mind. HG19 is an older reference file, reference genome file. So everything that you get here, it's okay because we're using RSIDs and those should be consistent between reference genome builds. But it's just something to keep in mind that the chromosome positions here are referencing the older build. So let's go ahead and check these 2 SNPs that are missing if we can find any suitable replacements for them. For this first SNP, if we check the VCF files, we see that this one is in perfect LD, which means it has a D prime and an R squared of 1.1.0, which means that every time this particular SNP gets inherited, this one will get inherited as well. This one. There's a couple of options that you can have here, which is why I've put this full table. But for the second SNP here, this is the one that's missing. This is in perfect LD with it, so it gets inherited with it each time. But you start seeing here that even though 0.99 is a very high number, it's not exactly 1. So there are a couple of options here. So we're going to go ahead and replace those 2 SNPs with the SNPs that we know are going to pass and get inherited with that in LD. So we extract the variants again. This is the same command, except now we've replaced the 2 SNPs with the SNPs that are in LD. We extract them the same way. We're using the same files. And now you see here when we inspect the log file that we find 90 variants. Great, now we can move on. We've got 90 out of 90. This is as comprehensive as we're going to get. So now we go ahead and move on to actually calculating the PRS. So again, the PRS leverages the beta effects that come from the large genome-wide association study, these weighted alleles, and looks at the genotypes of each individual and then assigns a score to them. So we're using Plink to do this. It reads in our abbreviated Plink file that we've made, our score path, the RSIDs that I highlighted earlier, and it makes a score file that we can use. Because there are 90 loci associated with Parkinson's disease, there are also rare variants that contribute to Parkinson's disease as well. They are incredible at predicting PD. Rare variants have a larger effect size and are more detrimental, but they are just that. They are also rarer. So we choose to exclude them for PRS. This goes back to the figure that we made earlier, and we will make later in this notebook. We are removing rare variants to see how the PRS might change for these individuals. And we'll compare the 2. So these are the rare SNPs that we're removing. This is also in tandem with the genetic resource manuscript I talked about in the slides earlier that we made available after the first release of ANTPD. These were the same SNPs excluded there as well. So these include SNPs like G2019S for LARC2, if you're familiar, the 3070 GBA SNP as well. So we remove them, and then we calculate PRS excluding these rare variants. So here we'd be expecting 83 because we've removed, we'd be expecting 83 variants. So to clarify, PRS 90 is including all of our variants. PRS 83 is excluding rare variants. So we're merging both of these files together. You can uncomment here, but this file, how it's put together is you will have each individual's ID, their PRS 90 score, and their PRS 83 score. And you can calculate the dosages. And then we start getting into the visualization. So the PRS 90 and 83 that we've made, the demographics information that we've also made. The genetic ancestry PCA, we've provided this as a part of the notebook. And then the transpose raw dosages that we've just calculated here, we are keeping the dosages in order to calculate PRS. So we're just merging the files here, making a table broken down by sex, age at baseline, education across the enrollment arms that we were looking at. We go ahead and plot the PRS distribution. So this is, again, PRS 90, including all the variants, PRS 83 after removing the rare variants. This is without, we haven't run a regression or anything. These are just broken down by the enrollment arms. And there you have it. That's actually, you could stop here. You have calculated your PRS. You've plotted it out. And this is the sort of distribution that you expect, where a bulk of the people fall into the middle. A couple people will be on the tail ends. We also plot the PCs to see if the principal components are actually capturing the variance that we expect it to. We run a few statistical tests here. These are just t-tests to see if the PRS scores between the enrollment arms are statistically significant. And you can see that between healthy controls versus positive Parkinson's disease cases, that yes, the PRSs are statistically significant and very much so, 8 times 10 to the negative 96. But you can also compare other enrollment arms as well. So for affected versus unaffected but genetic recruitment, you can see that the p-value is significant for after removing the rare variants, but not prior to that. And there are just a couple more examples. Here is an example of running a regression. Here we're running the ordinary least squares regression, OLS for short. OLS is a common technique for estimating the coefficients of linear regression equations, which describe the relationship between one or more independent quantitative variables and a dependent variable. Again, really want to remind you about the considerations at the top. This is not a complete model because it doesn't account for all the possible covariates. So these results are, take them with a grain of salt. We read in that covariate file that we made in our last notebook. We subset the scores earlier based on enrollment arm. We make a function just to convert the output of the regression. And here is us actually running the regression. So we are predicting for status. So here it's between if you have Parkinson's disease versus if you're a healthy control, using PRS 90, for example, and adjusting for education, sex, family history as our covariates. Again, not a complete model. It's just an example of one. These are the results that you get. This is an abbreviation of the p-values, the confidence intervals, both lower and higher, 95% confidence intervals, and the coefficient. And you can see here that the p-value of PRS 90, PRS predicts the status between Parkinson's disease and healthy controls, and it's quite significant as well. So you can modify this to your liking. You can really subset this to any enrollment arms that you want. You can play around between PRS 90 and PRS 83 after excluding rare variants. We're just visualizing the mean effect of the risk variance on the different recruitment styles. So we also output here each SNP, their allele, their beta, but also the frequency difference between two of the enrollment styles. So for here, just as an example, I've kept genetically enriched unaffected individuals. So these are individuals with mutations in those genes that we know are associated with Parkinson's versus healthy controls. So people that have no mutations in those genes and also exhibit no signs of neurological disorder. This is just a visualization to see the effect mean. And we also calculate to see which SNPs have the largest effect sizes. So for here, this particular SNP, LARC2's G2019S, GBA's 3070 and the TMEM 175. These three particular SNPs are having the largest effect sizes based on our analysis. So we go ahead and adjust and remove them, run our regression again, and we see that after adjusting for these variants, the PRS difference is between the two arms. So the unaffected individuals and the healthy controls, they're not significant anymore. So, and this is to be expected because the genetically enriched unaffected individuals might have mutations in these genes, whereas the healthy controls won't. And we save out our files. The files here that we're saving out are, just to recap, the SNPs here and the frequency differences between the two arms that you want to keep. And I haven't shown it here, but the last file that we're saving out here are IDs and their PRS 90 and PRS 83. So these are the PRS scores per person for everyone involved in AMP. And that is it.
Wow, thank you so much. Were there any questions regarding anything that was presented in either the first or the second notebook?
My name is also at the top of those notebooks. If you have any questions after you've gone through what I imagine are really, really dense PDFs, you're welcome to ask. Even though there are some unaccounted covariates, should we not see a higher difference between? Yeah, Alejandro, I do think that it's a matter of adding in more covariates. You can actually see the density plots. I do think it's also squished because I've kept each of the enrollment arms. Whereas I think if we only kept two of the individuals, we might see a more drastic difference, but it might be the way that it was plotted.
Thank you. Are there any other questions? Okay, I'm going to attach in the chat the link to the Google form. If you could just fill it out, it's a quick survey, just so we can better serve you with these webinars. And then let me just really quickly get back to my screen that I was sharing. There we go. Thank you. So once again, I sent over the survey link. So yeah, if you could take the time to do that, that would be great. And with that, were there any other questions before we move on? Anyone? Okay. Thanks again, Mary, for a stellar webinar and just stellar notebooks. And these are definitely stellar resources. And I really like just how simple you made everything. My background's in proteomics, so I felt like I could actually really understand what you were doing and how. And just all the annotation made it so easy to really understand what's happening in the notebook and in the code. So thanks again for all the hard work you put into it. Yeah, just really, really great work. Our next webinar is in about a month on September 14th. So if you're interested and you're not on the invite, please feel free to let me know and we can get you that information. With that, this is a list of everyone involved in AMPD. So, you know, thank you definitely to Mary, also individuals in the data analysis working group and just all the other working groups within AMPD. And with that, just thank you everyone for attending. Thank you for your participation and thank you in advance for filling out that survey. Once again, filling out the survey really helps us tailor these to, you know, present you with information that you find valuable. We do have 2 minutes left, so we can either head out early or if anybody has any last minute questions or comments. Does anybody have anything last minute to state? Okay, guess not. Okay, thank you so much, everyone. Thank you, Mary. Enjoy the rest of your Wednesday. Yeah, thank you.
Thank you,
All right,
Mary.
take care,
That was
everybody.
excellent.
Okay, bye.